Hi, I'm Yuxing. Thanks for inviting me to introduce our work. We are from Sun Yat-sen University in China, and this is a video about our paper, Gene-based message passing for drug repurposing. I will go through these several parts to introduce what we have done. So the first is topic background. What we want to do is drug repurposing, which means find a new indication for an existing drug, and it's a more efficient way than traditional drug discovery. In our work, we introduce gene to connect with disease and drugs. So we construct a heterogeneous network in which drug, disease, and gene are modeled as nodes, and the relation between them are edge. Intuitively, we believe that there is a relation between the drug and the disease when they connect with the same gene. But the mode of action of a drug and the pathological mechanism of a disease actually involve series of genes, not only one. So in the heterogeneous graph, these mechanisms can be presented as several paths from a drug or disease node to a series of gene nodes. We want to make better use of these paths. So the second part is our idea of gene-based message passing. Lots of graph neural network model is not suitable for encoding gene path information. As you can see in the figure A, when the data is a bigraph, in the graph there is only drug and disease. The red node is disease and the yellow node is drug. And we put the graph into a GCN model. Each node is updated by the surrounding homogeneous nodes. As you can see, after one iteration, the central disease node is updated by connected 3 drug nodes. It's reasonable because at each message passing process, the nodes are homogeneous. But if we look at figure B, when we add genes into the graph, the central disease node is surrounded by genes and drugs. The nodes in message passing process become heterogeneous. But message between gene disease or message between gene drug are not several triples. They're more complex. As I said in the last slides, the mechanism of drug or disease are presented as path in heterogeneous graph. We notice that the different information should be passed in different ways. Otherwise, the model cannot extract the useful knowledge in the graph. Like the gene path in GCN-based message passing will be cut into several triples. So in our work, we separate the message passing process on drug disease bigraph and on gene path. We employ long short-term memory LSTM to perform message passing along the path. The hidden state of the terminal node in the path is regarded as the message that aggregated all information of this path. That means in a path from genes to disease or drug, the hidden state of the drug or disease node can capture the information of all genes on the path. We choose 2 drug disease datasets from previous works, and we call it DD dataset. And we collected the related genes for drugs and disease, and also gene-gene relations from external database. We called them DGD dataset. And in the DD dataset, there is only drug and disease and the relation between drug and disease. In DGD dataset, there is drug, disease, and genes, and the relations between drug and gene also the relation between gene and disease, but there is no relation between drug and disease. Here is the pipeline of our framework GeneDR. We input DGD dataset to Trans-E to integrating the global information for every node, and the output of the Trans-E is the node initial embedding in our framework. So the gene path for each drug and disease are also extracted from DGD dataset by random walk. And then after LSTM-based message passing layer, we get the updated node embeddings with the information of gene path. Then these drug disease embeddings are used to initializing drug and disease nodes in the drug disease bigraph. The information in the bigraph is aggregated by GCN-based message passing layer. And to better use the data, the resulting embeddings at this process is back to LSTM-based layer to update around the workflow again. After 2 rounds, drug disease embeddings are connected together and input into a fully connected layer to output the final link prediction. We performed 5 cross-validation on 2 DD datasets. The details about the experiments can be found in our paper. And as the table shows, we compared our proposed GeneDR with several state-of-the-art methods for link prediction on 2 datasets. Our method performs the best among them. Turns out that GeneDR make better use of the gene path information. We also did the ablation studies to investigate the effect of the gene path and the LSTM-based message passing layer, and the results indicate that they are both important, and the LSTM-based message passing makes better use of gene path information, probably by simulating flow of message along the gene path. At the end, I'm grateful to all the co-authors who are my tutors and my teammates. Thanks for them. And there is another work from my tutor. I will make a simple introduction about his work on his behalf. Secondary mutational genes in yeast knockout strains show similar characteristics. Genome-wide knockout analysis is a powerful tool to reveal cellular functions of genes of interest. The yeast knockout collection, YKOC, includes over 20,000 knockout strains. The YKOC collection has provided a wealth of genome-wide functional annotations and understanding of cellular response to perturbation. Some studies explored the impact on genomic perturbation driven by deletions of single cells. The impact on genome evolution in eukaryotes driven by genome-wide deletions of single genes hasn't been systematically explored. To determine the consequences of single gene deletions, a recent study has sequenced the whole genes of most strains YKOC. The loss of individual yeast genes can result in secondary mutations. However, whether these secondary mutations are random or not remains to be elucidated. It's also interesting to examine whether these genomic perturbations are associated with gene expression perturbations in the same knockout strain. Analyzing the data above, we found that secondary mutational genes in the same YKOC strain tend to be in same chromosome and involved in the same biological process, shows evolutionary similarity. In addition, secondary mutational genes tend to be involved in the same biological process with differentially expressed genes in the same YKOC strain. These results provide evidence that secondary mutations fixed in YKOC strains are not random, which can be used to study evolutionary trajectory and the functions of deleted genes. Thanks for listening.